The antibody is generally consisted of the heavy chain and the light chain. The heavy chain is always the constant region, whereas light chains are composed of both constant and variable regions. The antigen binding sites are present in the light region, whereas the Fc region, which binds to immune cells receptors, are present in the heavy chain region. The antibody can be a single molecule, such as IgD, IgE, and IgG. Or they can be a couple of molecules connected together, such as IgA antibody, or a cluster of 5 antibody molecules connected together, such as IgM antibody. Now we will have examples of antibody functions. Firstly, agglutination. Here, the antibodies form a large complex with bacteria. This complex neutralizes the bacteria and facilitates their removal by phagocytic cells. Secondly, antibody-mediated phagocytosis. The binding of antigens to the antibody activates the complement system linked to the antibodies. The complement system in turn works as a navigator for the phagocytic cells to the site of the antigen-antibody complex. Complement proteins activate the phagocytes, which become eager to engulf the antigen-antibody complex and remove them from the blood. Thirdly, antibody-dependent cellular toxicity, where antibodies attack antigens on the surfaces of the targeted cells. Then, the Fc part of the antibodies binds to specific receptors on the natural killer cells. The natural killers do not phagocytose the antigen-antibody complex, but instead they release a group of perforating enzymes which ultimately destroy the target cells. This video is narrated by Hassan Nawab, medical student at King's College London.
The immune system depends on special binding molecules called immunoglobulins. These are also known as antibodies. Immunoglobulins are often referred to in shorthand as simply Ig. Igs are Y-shaped molecules which connect on one end to an invading microbe, which is known as an antigen, and then they connect on the other end to bind with various white cells. These specialized antibody molecules come in different shapes and sizes, which provide a great deal of flexibility in matching and destroying targeted antigens. Antibody molecules are found floating in the plasma as well as on the surface of specialized B cells. Immunoglobulins are constructed from two types of amino acid chains, heavy chains and light chains. Ig molecules contain 2 identical strands of heavy chains and 2 identical strands of light chains. The side of the Ig that has 2 heavy chains forms a stable non-variable receptor point called an effector. This effector is part of the Ig molecule that binds with our own immune system cells. On the opposite end of the antibody are 2 antigen binding sites, each with a light chain and a heavy chain combination. This combination of light and heavy chains forms highly variable tips that can adapt to specifically match a wide range of antigens. Immunoglobulins come in 5 basic types: IgA, IgD, IgE, IgG, and IgM. Each type targets a specific type of antigen, which determines the wide chain structure of each type of Ig. IgD, IgE, and IgGs all use a single wide chain, whether they stand alone or as receptors connected to white cells. IgAs use a double wide chain, which is called a dimer. IgMs are unique in that they form either as a single wide chain when expressed on the surface of a B cell, but the standalone version of the IgM molecule forms a combination of 5 wide chains, which is known as a pentamer. Consequently, standalone IgMs form a very large antibody molecule. In the next video, we'll look more closely at the functions of each of these 5 IGs.